It's my pleasure to introduce the last but not least speaker, Dr. Atocha Romero, who's a cancer biologist and director of the liquid biopsy lab at the Puerta de Hierro Hospital in Madrid. She's going to talk to us about the gut microbiome and its effect on neoadjuvant chemo-immunotherapy.
Okay, thank you very much for the kind invitation. It is a pleasure to be here and this interesting meeting with such excellent speakers. I'm going to be talking about gut microbiome and its impact on neoadjuvant chemo-immunotherapy based on the data that we have from NADIM-2 trial. So, we have to acknowledge that microbiome is gaining attention in the medical world. Indeed, we have more prokaryotic cells in our body than eukaryotic cells by several orders of magnitudes, and these command cells interact synergistically with the host to regulate many important processes, biological processes. The organ that has the highest number of microbes is the gut, and gut microbiome has been shown to modulate the immune response, to educate the immune system. And in relation to cancers, some anti-tumor effects has been linked into microbiome, and also some pro-tumor effect. And as I mentioned, so I'm going to be presenting some data regarding the NADIM-2 clinical trial, in which we did 16S gene sequencing, and also shotgun metagenomics and some metabolomic analysis. So, as I mentioned, several microbes have been linked to improve outcomes in patients treated with immunotherapy-based treatment. For example, Faecalibacterium provnicis has been shown to boost the efficacy of immune checkpoint inhibitors. In the same way, Akkermansia multifida has been shown to predict response to immunotherapy-based treatments in non-small cell lung cancer. And I think right now there is solid evidence supporting that gut microbiome is a strong modulator of the response of immune checkpoint inhibitors in solid tumors and in lung cancer. And I think that an effort that has been made to translate this information or make this information feasible and available in the clinical setting, the group from Gustave Roussy has recently developed a signature by identifying good bacteria and bad bacteria, and this signature can be potentially run very easily by qPCR, and it is apparently of prognosis significant and may help you, will provide prognostic information at baseline. So you all know about NADIM-2 trial. In this trial, non-small cell lung cancer with stage locally advanced disease, potentially resectable non-small cell lung cancer, were randomized either to neoadjuvant nivolumab plus chemotherapy, and in those patients that achieved an R0 resections were treated with adjuvant nivolumab, and then the control arm were treated with chemotherapy alone plus surgery. This trial was conducted by the Spanish Lung Cancer Group. It's an academic trial. This was a positive trial with increased pathological complete response, PFS and OS. You all know that. But I want to stress that the Spanish Lung Cancer Group, of course, besides demonstrating the benefits of the perioperative approach of chemo-IO, has made a lot of efforts to understand the biology behind the response to neoadjuvant chemo-IO, and by studying different biomarkers. So this is data from NADIM-1, in which samples, blood, plasma samples, and tissue samples were analyzed by different techniques, showing, the role of TCR repertoire, B cells, peripheral B cells, HLA class 1 class, and also more the classical biomarkers such as ctDNA, TMB, or PDL1. So being aware of the importance of microbiome, fecal samples were recruited in the NADIM2 trial. These were not obligatory, but we were able to recruit 81 samples, fecal samples at baseline, and also fecal samples were also collected after the neoadjuvant treatment, but before surgery in 54 patients. So these samples were analyzed by 16S gene sequencing and also by metagenomics. This is the panel in the upper right that we use for the analysis of 16S sequencing analysis. The panel also includes, of course, target the 16S gene, but also includes targets other regions outside 16S for a small, reduced number of bacteria. So some of the parameters that we have analyzed is the alpha diversity, which is the intrinsic diversity within the samples. There are several indexes that we can use to measure this alpha diversity, but basically what we look is at richness, the number of species, and also the evenness or the distribution of these populations within the samples. So regarding the results, basically we do not see a difference in baseline microbiome between controls, the control and experimental arms. So we can assume that the controls and experimentals were balanced in terms of basal microbiome. In the same way, there were no difference in alpha diversity between experimental and control and patients in the control arm. And we don't see, so we have to take into account that this is very small numbers for microbiome analysis, but this is the numbers that we have for the NADIM2 trial, and we do not see a clear effect of changes in diversity due to the treatment. But when we split and we analyze richness, we do see that there is an increase in richness in both arms when we analyze pre and post fecal samples, and this is something we want to confirm with metagenomic data. And regarding evenness, just to remind you that evenness measures how the species are distributed across the sample. So in a sample in which the species are well balanced, are homogeneous, evenness tends to 1, whereas when the distribution is unbalanced, evenness tends to be 0. And so we do not see difference between experimental and control arm in evenness. But we do see an effect in evenness when we compare pre and post neoadjuvant samples. So that what we see, despite the low numbers, is that evenness decrease in the experimental arm. So chemo-IO would potentially promote more unbalanced microbiome in patients treated with chemo-IO, and this is not seen in the control arm. So now focusing on what is happening only in patients treated with chemo-IO, there is no difference in alpha diversity when we compare patients who achieve a pathological complete response versus those patients who did not achieve a pathological complete response. This is something you can see in the upper left panel. And also when we compare pre and post neoadjuvant samples, what we see is that there is a decrease in alpha diversity in those patients who do not achieve a pathological complete response. So when we now split and focus in what is happening regarding to richness and evenness, again, we see a trend, not significant, but a trend in an increase. So chemo-IO will potentially promote an increase in richness, and although this is not significant in both PCR patients and non-PCR patients, but this effect that I have told you before, that we see that this decrease in evenness, this promotion of a more unbalanced microbiome that occurs only in the experimental arm but not in the control arm is driven by patients that do not achieve a pathological complete response. It's only, these changes are only, significant, as you can see in the lower right panel, in patients who do not achieve a pathological complete response. Of course, this is, we have low numbers, and we need to confirm this, hopefully, with more sized cohorts. So, as I mentioned, basal diversity, alpha diversity does not predict tumor response, it's the changes. And so this is also true for survival data. So basal diversity does not predict survival data, but we do see, especially when using the Chow index, an effect in the control arm. So patients with higher diversity in the chemo alone group would have improved outcome, and this is significant despite the very low numbers. And this effect is not seen in the experimental arm. So regarding the genus that we found, we can confirm that Akkermansia is associated with improved outcomes, and also with Erythrococcus and Ruminococcus. And you can see that the distribution of these bacteria in our population and in non-PCR patients and PCR patients. So we classify the patients for this differential abundance analysis in PCR patients versus patients that do not achieve a complete pathological response. We also found that Bifidobacterium, although was not associated with pathological complete response, it was associated to survival outcome in our cohort. And we also found some, but this is very low numbers, some genus associated with colitis, immune-related colitis, but this is something we may need to confirm. So regarding the SAT-GAN sequencing analysis, we have analyzed 81 samples, stool samples by using SAT-GAN metagenomics using the Illumina platform. One sample from the experimental arm failed, the QC, and so it was removed from the analysis. So when we do metagenomics, we need to get rid of the human reads. But if we take into account the human reads, we can see that it's very homogeneous across the whole cohort, and there are some high reads, some outlier reads, and all these high outliers, high human reads, were all in patients who did not achieve a pathological complete response. And this difference were significant for the experimental and the control arm. So meaning probably this desquamation or inflammation, that could be a surrogate of desquamation and inflammation correlating with poor treatment outcome. So now at a species level, we again confirm that Akkermansia is a good, is associated with a pathological complete response. Also Coprococcus comes, which has been also identified. It's in the topo score, the signature that I mentioned at the background also was associated with response, and some other bacterium, like Eubacterium serum, that has been linked to worse outcome, we also identified. And we have also identified some species correlating with immune-related colitis, but this is also small numbers, and we need to correlate this or validate this in more sized cohorts. And we have also identified several pathways that, functional pathways that are related to, associated with pathological complete response. Some of the pathways that we have found is the L-cysteine biosysteine from L-methionine. L-cysteine is a precursor of the glutathione and hydrogen sulfide, and it enhances redox balance, reduces inflammation, protects epithelial cells, and this may suppress the immune response and promote mucosal tolerance, and promotes immune tolerance and reduce pro-inflammatory signaling at the gut level. We have also found significantly the production of butyrate. And this is something we would like to explore in more detail. This is a study from a Japan group in which a retrospective study of non-small cell lung cancer in which patients were treated with immune checkpoints inhibitors, and also the patients had this probiotic that apparently is very common in Japan for treating dysbiosis. And what they found is that patients that received this probiotic had improved survival outcome. In a clinical trial for metastatic renal cell carcinoma that was presented in ASCO a couple of years ago, and this is the paper, the same bacteria, the same Clostridium butyricum bacteria, that is a bacteria that produces butyrate, was administered to this cohort of patients, and despite the primary endpoint of the trial that was to characterize the effect of these agents in relation to the abundance, the changes in the abundance of gut microbial populations, this primary endpoint was not made, but the authors clearly saw that there was an improvement in survival outcomes and also treatment response. We have also seen this pathway of mycolate biosynthesis and fatty acid biosynthesis significantly increase in PCR patients, and this, as you may know, mycolic acids are presented in the cell wall of the bacteria to immune cells in a similar way that PD-L1 is presented to PD-L1 and promoting immunotolerance. So we want to explore more details these pathways. And finally, we have also performed some metabolomic analysis from the baseline plasma sample in NADIM2. So from the 86 patients that were recruited in the NADIM2 trial, we had a viable 81 samples. We have to take into account that we did ctDNA, many biomarkers, so it is important to know that you need very few amount to do this kind of analysis, and we have analyzed 121 polar metabolites and 69 non-polar metabolites, and we did this by using chromatography coupled to mass spectrometry, and we have found several metabolites that have been associated with improved with PCR, and some metabolites that are associated with like bad metabolites in the right side. But because the high number of metabolites that we were analyzing, the only metabolite that resulted significant after Bonferroni adjustments was the histamine, which was significantly increased in patients who achieve a pathological complete response, and this is the plasma histamine levels, the box plot showing the plasma histamine levels incomplete in patients who achieve a complete pathological response versus those who did not. And this is also true for survival outcomes. So these are the Kaplan-Meier curves for relapse-free survival and overall survival according to histamine levels in plasma. As you can see, patients with high histamine levels in plasma, there were no deaths in the experimental arm. So, of course, NADIM2 trial was not designed to study the microbiome, and so we have these small numbers, but we may be able to validate or improve or increase the number of samples to be analyzed by with this study also promoted by the Spanish Lung Cancer Group, and whose PI is also Dr. Provencio. The main objective of this real NADIM study is to evaluate the prognostic value of ctDNA, of the clearance of ctDNA, but we have also, there are several exploratory objectives to study TCR, VCR, and doing spatial transcriptomics and so on, and we have already collected 114 fecal samples, and hopefully we can validate the results that we are presenting here. So basically, the results that we have so far is that neoadjuvant chemo-immunotherapy may alter the gut microbiota by increasing the richness of species and reducing evenness, with lower evenness being potentially associated with poor pathological response. We confirm that the abundance of some well-known bacteria associated with improved outcomes, such as Akkermansia multifila and Coprococcus coccons, are beneficial commensals. And we also have seen that plasma histamine levels are associated with pathological complete response and survival outcomes in Nadine 2 patients. This is at baseline, and only in this effect is only seen in the experimental arm, but not in the control arm. We are also going to start this migrant trial in which patients will be randomized to fecal microbiome transplantation versus just the standard neoadjuvant durvalumab versus chemotherapy, and then surgery, and then adjuvant durvalumab. And the objective is to evaluate the efficacy of this FMT in these populations. And donors will be patients from Nadine and Nadine 2 who achieve excellent survival outcomes. And just thank you very much for letting us the opportunity to present our work. Thank you very much. I'd like to thank all the people in the lung cancer group at Hospital Puerta de Hierro, and to Roberto and Alex, they're sitting there, that are who were the people involved in this microbiome analysis. And thank you very much for your attention.